We will move on to the final speaker of today, actually, before our VC panel after dinner. Kristen Fortney, are you there? We can see you on the screen. Can you hear us?
Yes.
Great. So whenever you're ready to give your presentation, we very much look forward to see it.
Great, thanks. I'll pull it up right now. Hello, everybody. It's great to be here today. First, thank you to the organizers for having me. It's a wonderful conference. And today I'm going to tell you about BioAge. So we are a clinical stage biotechnology company building a pipeline of therapies that target the molecular causes of aging. And what's unique about our approach to aging is our focus on longitudinal human data sets collected over decades of aging. And what we do is we have several biobank partners that up to 50 years ago started collecting samples from healthy biobank participants. And those same people were followed up for the rest of their lives, so over 50 years of aging. And fundamentally, what we do as a company is we go into those samples, we enumerate all the molecules in there with modern technologies, proteins, metabolites, transcripts, and ask the question like, what's changing with aging? And also importantly, what predicts future health span and future lifespan? What's different about the molecular profile of a 50-year-old who goes on to live 90 plus in really good physical and mental health versus the rest of us? And how can we target those differences? So that's how we build our map of all the pathways that are correlated with human longevity, human health span. And then from that point on, we're very focused on the practical. So there's, as you know, there are many different dozens of targets that are important for longevity. And at the company, we are focused first on those that are nearest the translation. And what that means is that for several of these pathways, there is already a clinical stage asset out there, a drug that's been in a phase 1 study that's known to hit its target, that we believe is going to have an impact on aging. So we're focused now on bringing in licensing multiple of these drugs and advancing them to human trials for proof of concept in aging. We've brought in 3 of those programs so far, and we plan to bring in several more over time. So today, I'm just going to briefly introduce our approach to drug discovery and development at BioAge, and then do a deep dive on one of our lead programs, which is focusing on immune aging, where we're currently in the middle of a phase 2 clinical trial. So briefly, this is why we work on aging. I think that this audience understands this very well, but aging is the root cause of many severe diseases. Diseases like Alzheimer's, heart disease, for the most part, they do not happen to 20-year-olds, they happen to 80-year-olds. And we believe that by looking at all of these diseases through the lens of aging, we're going to find important new causal targets for them. So just a brief overview of BioAge, we are developing transformational disease-modifying therapies from our innovative data sets. And as I mentioned at the beginning, what's unique about our approach is this really target ID platform, this computational data-driven target ID platform built on top of our unique longitudinal biobanks spanning 50 years of human aging. We're focusing first on in-licensing de-risked clinical stage assets for those pathways that are emerging from our map. We brought in 3 of these so far with a plan to bring in many more. And we've raised to date about $130 million from great investors like Andreessen Horowitz and Kaiser Foundation to support these programs. And I'm sure, you know, all of the companies that are in the aging space have to think about what does it actually mean to develop a therapy for aging. And this is how we think about it at BioAge. So we are betting on mechanisms, on biology that we think ultimately has the potential, like these therapies, to help a very large fraction of the aging population and go to an aging indication, something like frailty, that's very highly prevalent, where there's high unmet need and also huge market potential. But because those clinical trials are challenging and are going to involve a lot of people, we are also first going to, for each of these mechanisms, going to what we're calling a model indication. So a first indication in the clinic where we can run a very efficient phase 2 clinical trial that will serve as a proof of concept for aging, but that also is a well-defined disease indication in itself. And I'll explain what that means for each of our 3 programs as we move along. And just briefly, what I think is the driving force behind BioAge is our focus on human aging. So as you all know, there are all these natural human experiments going on all around us. And I've always believed that the low-hanging fruit in aging biology is going to be to copy what's already working. And what I mean by that is that there already are humans out there who are living, 100 plus with really good physical health, really good mental health. What's different about their biology? And can we mimic that in a way that will help out the rest of us? And this is just showing you here a distribution of the age at death of the people in one of our cohorts. And you can see that it's very spread out, right? So that means that by looking at the molecular differences here, we can begin to explain what's different about the biology of people who live, 100 plus in great health versus those who only make it to 60. So the BioAge platform that we've built over the last 5 years now is human first and computationally driven, focused on portfolio creation. And this is sort of the workflow that we go through. We start with our source, which is these human aging cohorts with decades of follow-up. Longitudinal patient samples collected to detailed health records that have information not just on lifespan, but also health span. Step 2 is we characterize. We use multi-omics technologies to find the different functional pathways and see how they change with aging. 3 is map. Our computational network mapping identifies targets that are correlated with the outcomes of interest, lifespan, and health span. 4 is validate. So for any pathway that we're excited about, and for a molecule out there, a drug that we're excited about, we bring it in-house and we test it in animal models of aging to see if it affects the phenotype in the way that we predict that it does. And then 5 is build. For a molecule that we validated internally that we believe is going to affect human aging, we bring it in and take it forward to a proof of concept in the clinic. And just to give you an overview of where we are today, we have over 1.5 million clinical data points from our partnerships, over 65 million molecular data points from the assays we've conducted, yielding 50 million molecular relationships. And in terms of the company, we're assessing about 20 pathways per year. And so far, we've bet on 3 clinical stage assets. We're evaluating another 10, and we have 1 discovery stage asset at an earlier stage of our pipeline. The reason we've built our strategy this way is because we're really de-risking development at every step. So we know that our mechanisms will be relevant in humans because we're starting with human biology. We're starting with human signal. 2, we believe that these high throughput experiments, omics technologies, will really maximally sample the important biology. 3, the systems biology and network analyses we do help us identify highly networked targets and their neighbors that are more likely to be relevant to disease. 4, we use, for all of our evaluations in animals, we use naturally aged mice because we believe that these give superior disease recapitulation versus knockouts. And 5, we think it's really important early on to bring these molecules into human trials and get that first peek of what they're doing in actual humans. And just to give you a brief overview for one of the most recent drugs that we brought in. So just a few months ago, we brought in an apelin receptor agonist from Amgen, and a very high level overview of how this fits into our workflow. So importantly, one of our focus areas at BioAge is on muscle aging. And our cohorts have a lot of information relevant to that, including how grip strength changes over time, how walking speed changes over time, various measures of frailty. And 2, our proteomics data let us build sort of a subnetwork that's very relevant to apelin signaling. When we went and looked at that network in our cohorts, we found very strong relations between the network and between apelin itself and multiple key functional outcomes, including prediction of probability of survival with full mobility. So you can see in that little diagram there that people who have more apelin signaling are living longer and are living healthier. And we're submitting a manuscript soon with all these data. Then when we got excited about this molecule, we brought it in from Amgen. We put it into multiple different mouse models of frailty and impaired muscle function. And that's just one of the pieces of data right there, showing that, so usually if you have an old mouse and you cast its arm, you immobilize it, they get substantial atrophy. But as you can see here, the mice that were treated with BGE 105 the apelin receptor agonist, are protected from atrophy. And we're really excited about that because there's a lot of clinical indications where older people are in hospital for a long time and suffer from atrophy that they can't recover from. And finally, based on these data, we in-licensed this drug just a couple months ago. So very briefly, we now have 3 different therapies that are clinical stage. The one that we're going to zoom in on today is BGE-175 for your immune aging. Now, as you know, immune aging is really important for a lot of different diseases. Older people are susceptible to, they don't respond as well to vaccines. They're more susceptible to pretty much any viral or bacterial challenge, which we see every year with the flu, which we're seeing right now with COVID-19. So here we're initially going to develop this molecule in the clinic for COVID-19, but that's sort of our immune proof of concept for a bunch of other indications that are driven fundamentally by immune aging. And in terms of the target, this molecule is a very potent inhibitor of prostaglandin PGD2 DP1 receptor. BGE-117 is a very potent inhibitor of HIF-Prol hydroxylase hypoxia-inducible factor. So it's basically activating your hypoxia pathways. And this one we're going to clinically develop first for anemia as in older people on the road to broader aging muscle indications like frailty. And similarly for the drug that we just brought in from Amgen, BGE-105. So this one also we think is ultimately going to be relevant to really broad aging indications like frailty. But the first proof of concept indications are going to be disuse atrophy, things where you see muscle atrophy happen just as we saw in the mice. Now, for the rest of the talk, I'm going to focus in on BGE-175, our molecule for immune aging. And as I mentioned, we're right in the middle of a phase 2 trial. So this is one of our most advanced programs to date. So just some highlights of the molecule. We believe that this molecule, because it's correcting certain aspects of immune aging, has really broad potential for respiratory viruses, bacteria, and viral bacterial co-infections, a lot of potential aging indications where we think this could improve outcomes. Now, a few facts about the drug. It's an oral small molecule. It's just a pill you could take every day. It already has safety data in over 2,400 patients from the original indication. So we know it to be a fairly safe mechanism. Interestingly, and I'll show you the data for this, it appears to have a dual acting mechanism. So as you know, a whole bunch of things go wrong with your immune system as you get older. And this appears to correct 2 different ones. So one thing that goes wrong as you get older is your dendritic cells don't migrate as well as they should in response to a virus. And that's partially due to the upregulation of this receptor. And also separately, you get too many infiltrating neutrophils in response to a virus when you're older, which leads to damaged lung tissue, and this can reduce that as well. We're excited about this molecule because it has the potential to be effective at any stage of disease progression, early or late. And of course, we expect it to be synergistic with antivirals with low risk of resistant selection, because it's not an antiviral, it's actually correcting a host mechanism. Our longevity map shows that this pathway is very dysregulated with aging, and we've generated very robust preclinical data. We've taken older mice, challenged them with lots of different viruses, including SARS coronavirus 2, SARS coronavirus 1, influenza, RSV, and also bacteria. And we currently have an ongoing COVID-19 phase 2 trial in the US, Brazil, and Argentina. So the key advantages of BGE-175 for COVID-19. So we decided, about a year ago to go into this patient population first because there's so much need and the mechanism has a lot of potential there. And as you know, COVID-19 is really a disease of immune aging. If everybody out there had the immune system of a 20-year-old, there would be so much less mortality and morbidity on a global scale. You can see on the top left there, the death rate is just so much higher in older people. And despite that, very few of the therapies being tried in the clinic are actually addressing this, right? They're not based on mechanisms relevant to aging. And in many cases, older people are actually excluded from trials, as you can see in the reference on the bottom left here, because it's hard to help them out. So we believe this mechanism can improve, that it directly improves human aging, will benefit older, vulnerable patient populations that are not currently served by existing therapies. There's also, importantly, a really strong safety profile across many patients in multiple trials. And as I touched on here, there's two different aspects of immune aging dysregulation, which this mechanism can improve. One is dendritic cells. So you can see on the left here that as mice get older, if you challenge them with virus like SARS or influenza, their dendritic cells don't migrate as well as they should. And two is the infiltrated neutrophils on the right. So I'm going to show you some data now with our drug. And for all of these data, we collaborated with Stanley Perlman, who's an expert in coronavirus models. He's at the University of Iowa. He's also a member of the FDA advisory committee that approved the Pfizer vaccine. He's been working on coronaviruses his entire career, and he developed several of the mouse models that other labs used. So together with Stanley, we've looked at models of COVID-19, at models of SARS coronavirus 1, and also mechanistic studies. And there's just a quote from Stanley on the bottom right here, that this age-dependent increase in DP1 signaling contributes to worse outcomes in the context of mouse infection with several different human pathogens. And our drug, BGE-175, might help counteract deleterious immune changes that occur with aging. So briefly, I've touched on this already, but we believe that treatment with BGE-175 will directly inhibit DP1 signaling. And this will, through a direct effect, because this receptor lives on dendritic cells, it will inhibit, it'll improve the dendritic cell activation and migration. And indirectly, it'll also lead to reduced neutrophil infiltration, which is apparently upregulated with aging. So here's the data. So now here we're looking at, I'm going to show you basically two different models of COVID-19. And this is the mouse adapted COVID-19. And we're looking here at middle-aged mice. So they're about 8 months old. And the way that the assay works here is that at day 0, they're being given a lot of virus. And at day 2, they start to receive our drug. And that at day 2, these animals, it's important to say, are actually quite ill already. They're already starting to lose weight. And this model is very challenging to rescue. And you can see here that in animals on vehicle, none of them survived, 0 of 6. And the animals on drug, 7 out of 8 survived. So nearly all of them. This went along with a 10-fold reduction in the viral titer, which you can see on the right here. And again, this molecule is not a direct-acting antiviral. It's just helping your immune system better clear the virus on its own. We looked in a second model of COVID-19. So this is the humanized mouse with the human ACE2. And again, nearly identical data. All of the mice that were vehicle treated died, but the mice that received our drug, nearly all of them survived, 80 to 90% of them. And again, this went along with a 10-fold decrease in the viral titer. And now we went and looked at a completely different coronavirus, so SARS-CoV-1. And as you can see here, this is the SARS from a decade ago. And again, you see the very same data where you see a full rescue from the virus for the mice that are on our drug. We think this is really important because it demonstrates that if the coronavirus continues to mutate, this won't be relevant for this mechanism. I think I'm getting a bit short on time, so I'm going to speed up. So just briefly, we can confirm mechanistically that our drug will indeed, improve the migratory ability of dendritic cells, and that it helps explains the rescue that we see. These data are all being reviewed right now and will hopefully be published soon. And we are right now in the middle of a COVID clinical trial, phase 2 trial in the US, Brazil, and Argentina, where we're looking at patients over the age of 65 who are hospitalized. So they're already, quite sick, but they haven't yet progressed to ventilation. And we're looking at that as the primary outcome. We're going to look at see if we can rescue subjects from progressing to respiratory failure or to death, just with 14 days of dosing with this molecule. And of course, this is just the starting point. What we see with COVID now, we see with influenza every year, the same kind of death curve. And that's true for pretty much any viral or bacterial infection. So we're really excited about the breadth of this mechanism. So that's just one example of the kind of program that we're developing. And our mission is to develop a pipeline of treatments that extend healthy lifespan by targeting the molecular causes of aging. And I'd just like to end with an acknowledgement slide. We've got a really great team with expertise in aging biology, AI, and drug discovery. Just a couple of weeks ago, about half of the team got together for a sailing trip, had a lot of fun. And I wanted to mention too, we are hiring. We have a lot of open roles, about 10. You can see them all here at this link at thelever.co/BioAge. Many of them are remote optional as well. And they include scientists, data scientists, and also clinical team and operations. And with that, thank you. I'm done.
Thank you so much. Great talk, Kristen. Really fantastic work you're doing. Do we have any questions in the audience? People are very eager to get some dinner, I think, here in Copenhagen. We have a question online. I have a question online. I'm writing great talk. How many individuals are in your cohort, in your long-term
Oh, a few
cohort?
thousand, several thousand. Yeah.
Several thousand? Okay, that's nice. Fantastic. Any other questions? We have a question from James Payer here. Can we get a microphone? Where is, we need someone running. He's coming. Exercise. You will never grow old.
I'm missing the room view.
Yeah. I apologize. You have to settle with me. Someone is running. There's a microphone being fixed. There are 2 microphones. One is working. The other one is not working. A microphone is on its way now. And we have a microphone.
Is this working? I think we've got it. Hey, Kristen. So we've been talking a lot today about biological clocks and biomarkers of aging and all of this kind of stuff. So with the clinical studies that you're doing at BioAge, how are you going to try to use the clinical work you're doing to kind of lever into this biomarker and aging clock story?
That is a great question, James, because we're doing these very short clinical trials, but at the same time, we're hoping to learn if these therapies are helping aging more broadly. So what we are doing in all of our trials is we are doing, full proteomes pre and post treatment. In the longer duration trials, people will have wearables on as well. But I'm hoping that with the proteomics, we can actually compare it to these signatures of mortality and longevity from our cohorts, as well as to everything that's published out there, and see if these therapies are not just, helping with this particular disease, but also ideally slowing aging. Yeah.
Sounds fantastic. I think we will stop now. We are running a little bit late and people are hungry. But thank you so much, Kristen, for a fantastic talk. Really look forward to what's going to happen in the future. And we'll see everybody in about 32 minutes. Thank you. Let's give Kristen another round of applause.
Thank you.